Okay, let's get back to it. So we're going to be starting the series of lectures addressing enzymes. And this is going to be a 3-lecture series because there's a lot of material to cover in here that I wanted to really be able to delve into. The first lecture here is going to be focused on kinetics. So a little bit about what enzymes are and some of their categorizations, but predominantly kinetics, how we evaluate kinetics and how kinetics are affected by different modes of inhibition. Then we'll spend some time looking at enzyme function mechanistically in the next lecture, and then the following lecture will be on the short side, and it will address the regulation of enzymes biologically. And that'll poise us really well to jump into next semester where we use a lot of these tools. So this first slide is addressing some of the primary questions that we want to be able to answer surrounding enzymes and kinetics. So what characteristics, features define enzymes? What makes an enzyme? Can the rate of an enzyme catalyzed reaction be defined in a mathematical way? The answer is yes. And these different models will give us insight into how these enzymes function. What equations defined the kinetics of enzyme catalyzed reactions? And we'll spend quite a bit of time with that math-oriented component. What can be learned from the inhibition of enzyme activity? Quite a bit. And what is the kinetic behavior of enzymes catalyzing bimolecular reactions? So we have, and that's going to be at the very, very end. We'll also talk a little bit about specificity and potentially get to some of the type of enzymatic reactions that take place in RNA versus just proteins. Okay, so we've talked about this a little bit, but it's worth really focusing in on now. Virtually all reactions that take place biologically are mediated by enzymes. And this is because we lean biologically towards chemistries which have high free energies of activation. Therefore, they don't take place rapidly under normal biological conditions. And this is really important because it gives us that control that's so important. Let me grab a pen here. It gives us that kinetic control over a process that, although maybe energetically favorable, as shown here with the oxidation of glucose, they take place very, very, very slowly because of the large barrier that the reaction has towards going to completion. And this gives us control over whether or not it occurs, and also gives us control over the rate by modulating the efficiency or activity of the enzyme that's doing it. So we can turn things on, off, or we can turn the valve up or down a little bit. And this is incredibly important in terms of us operating efficiently. So if we take a look at this diagram, you can see that glucose is being oxidized and takes 6 oxygens per unit of glucose, and if it happens perfectly, we get 6 equivalents of carbon dioxide and 6 molecules of water. Now, this is an energetically favorable reaction. You can see that the products have a much lower free energy than the reactants. However, without the presence of an enzyme, we have this very large barrier, free energy of activation, associated with the activation of the reaction. Now, in the presence of the enzyme, however, we reduce that barrier to activation. And that's the mechanism by which enzymes generally function, is they reduce the energy of activation. We're not increasing or decreasing the level of energy of the reactant. We're not changing anything associated with the products. We're just changing that intermediate through chemical intervention. So, what characteristics define enzymes? When we're talking about an enzyme, what kind of language can we use to distinguish its function from other enzymes? Catalytic power is a really important term that we can use to discuss an enzyme's function. And it's the ratio of an enzyme's catalyzed rate of reaction to its uncatalyzed rate, which is pretty straightforward. How fast can the reaction take place without an enzyme present? And how fast can it take place with the enzyme present? What is that ratio? How much does an enzyme speed up the reaction? Characteristically, we also talk about the specificity in terms of an enzyme's function for a substrate. Some enzymes can catalyze multiple substrates. Some have specificity for single substrates. And what's very important is that enzymes are highly specific. They're incredibly specific to their target substrate or substrates, so that there's very, very, very low potential for side reactions to take place with any kind of significant production. The regulation of enzymes activity ensures that the rate of metabolic reaction is appropriate. Talked about that before, but it gives us control biologically over the chemical events taking place in our metabolism, which is essential for life. You take this away, we don't have control of our chemistry, we don't have control of our biology or metabolism, and that just falls apart rapidly. There are a couple ways we can name enzymes which give us clues into what kind of functions they perform. We'll spend a little bit of time on that. And there's also sometimes really important coenzymes or cofactors, which are non-protein components which are essential to an enzyme's function. Sometimes these can be metallic, sometimes they can be small organic compounds. Regardless, in the absence of them, the enzyme cannot function correctly. So let's talk about this first one, catalytic power. Enzymes are incredibly efficient at accelerating reactions. Although they don't change the energy of the reactants or the products, or the energy released in a reaction, they dramatically change the rate of reaction by reducing the barrier to activation. And this number is pretty spectacular. 10 to the 21st power over uncatalyzed reactions, as much as. They're not all that efficient, but that is incredibly fast. And this is a good example. Urease is got a catalyzed rate of 3 times 10 to the 4 molecules per second. Uncatalyzed, very, very, very slow, and therefore the catalytic power for this particular enzyme is 1 times 10 to the 14th. You can see really quickly, without a catalyst present, negligible reaction. With catalyst present, about 30,000 reactions per second, and that's a significant improvement. So, specificity. Enzymes are remarkable in their ability to select the proper substrate over other molecules, to the point where we see selection for prochirality in enzymes all of the time. Even though chemically we can't isolate that prochiral component in a test tube or in organic chemistry laboratory, the enzyme can do it elegantly and with very, very high certainty. Enzymes also have the advantage of producing very high yields. Excuse me, This says above 95%. Typically, it's much better than that, much, much better than that. And that's really important because a lot of our biological chemistries take place in series of reactions, and these deltas that are not 100% really add up quickly. And we're going to have a figure on that in just a minute. And then last point, in terms of specificity. The specificity is controlled by the structure. The unique fit of the substrate with the enzyme and the interaction between the two of them is what dictates the chemistry. And we're going to get down that path in quite a bit of detail further in the lecture. And this is really important because we also get unique chiral centers produced in enzymes, whereas in a laboratory, a lot of our reactions form racemic mixtures, and we don't want racemic mixtures in our biological systems. We want specific, appropriate compounds to be formed. So this is a good example of a series of reactions that take place in classic metabolism. This is the conversion of glucose to pyruvate as part of our primary metabolism. And you can see here we have 10 different reactions that take place converting glucose to glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6 bisphosphate, etc., all the way to pyruvate, and this feeds into the TCA cycle. Now, this is classic glycolysis. Now think about this. What if each of these reactions was only 90% efficient? So 90% efficient here, 90% efficient here, 90% efficient here, etc. Well, that 10% non-specific yield accumulates spectacularly over the course of 10 reactions. And we go from being 90% efficient in each step to only being 35% efficient over the course of the conversion of glucose to pyruvate. And that is an unacceptable yield. Now, if we were doing this in an organic laboratory over 10 mechanisms, we'd be pretty impressed with ourselves. If we could get 90% yields in an OChem lab over and over and over and over, and in a long, complex synthesis with stereo specificity, have a 35% yield, we'd be pretty patting ourselves on the back. But biologically, that's absolutely unacceptable. That's a huge loss of energetic efficiency. We need that glucose to become pyruvate. So that's a really good rationale for why enzymes are so important for efficient biological function. So in terms of nomenclature, I'm not going to stress this significantly, but it's important to know that despite having a lot of scientific conventions which are not sensical, the nomenclature surrounding enzymes makes a lot of sense because the names are pretty descriptive as to what is happening. So in this case, we have the simple chart, which is just showing you a couple different classes of enzymes and talking about what they do. So example 1, oxidoreductases. They are redox reaction mediating. Transferases, they transfer functional groups, whether it's a methyltransferase, hydroxymethyltransferase, carboxytransferase, acyltransferase, glycosyltransferase. You guys get the idea. 3, hydrolase. It's a hydrolysis reaction mediating enzyme. Lyase, bond cleavage. Isomerase. You guessed it. It isomerizes stuff. So, lastly, ligase. Formation of bond with ATP cleavage. So, that one's not as obvious, but the others are very obvious. So, just keep in mind when you're reading the name of a enzyme, it's going to give you some insight into what it does. Now, we also talked about coenzymes. And once again, we're not going to spend too much time on coenzymes because we're going to come back to them in the lecture pertaining to regulation of enzymes in more detail. But this is a good time to just talk about some of the different types of cofactors that are commonly important in enzyme function. So, coenzymes and cofactors are non-protein components which are essential to enzyme activity. And this can be in the form of a metal ion. Whoa, I'm dragging stuff all over the place. I thought I tapped it, but there we go, metal ions. And then we also have coenzymes, which are non-metal, non-protein, small organic compounds that act as coenzymes. So, in terms of metal ions, you commonly see iron, copper, zinc, magnesium, manganese, potassium, and to a lesser extent, nickel, molybdenum, and selenium. It's not unusual to see, for instance, iron, absolutely essential for function in heme groups, right? And you can see this, catalases, peroxides, cytochromes, DNA polymerase. Without these low levels of metals, a lot of our biological functions don't happen at all. And then we also have these more complex coenzymes like biotin and coenzyme A and nicotinic adenine dinucleotide, or NAD. And you come here, this is NAD or FAD for the flavin adenine dinucleotide. And no, you don't have to memorize this. Please don't go and memorize all these. But be aware that this is what we're talking about when we're talking about a coenzyme or a cofactor. Now, let's dive in to the analysis of how an enzyme catalyzed reaction is taking place and how we can model it with math so we can get a good grip on how it's functioning. This whole study of science concerning the rate of a reaction that's enzyme mediated is or not is called kinetics. So we've done kinetics. We did kinetics in Chem 106 quite a bit. And what we're going to see here is not that different from what we did in Chem 106. In Chemistry 106, we did a lot of these same equations. So the only difference is the model we're applying them to is substantially more complex than the ones we used in 106. But it's just 106 on steroids, that's all. So don't stress too much. So enzyme kinetics, the goal of it is to determine the maximum reaction velocity an enzyme can attain and the binding affinities for substrates and inhibitors. That's really it. How fast can this reaction go, and how well does the enzyme interact with the substrate or various inhibitors as part of reaching this maximum velocity. If you know these things, you're 90% of the way there. And why do we lean on the math so much? Well, what it really comes down to is that the analysis of enzyme rate gives us insight into the mechanism and metabolic pathways associated with that enzyme's function. It gives us ideas onto how it's functioning and how it's regulated. So here's a couple terms that'll be really important for everyone to know and understand and be able to answer questions around. We need to be able to understand concepts of rate or velocity. We need to know about the rate constant, the rate law, the order of a reaction, and the molecularity of reaction. So, moving right into rate, the velocity of reaction or rate of reaction has to do with the speed at which the concentration of a product forms. So, in a reaction in which some reactant A is going to the product P, if you just look at the rate of change, of accumulation of P, that is our rate of reaction. You can conversely look at the rate of disappearance of A, since this is a single-step reaction. What's really important to note here is that the rate of this particular reaction is proportional to the concentration of A. Excuse me. So this is just a simple reaction of A to P, and it's first order because it's only dependent on concentration of A. This is also called a unimolecular reaction. So go here, unimolecular, first order. Now, if the rate depends on both the concentration of A and B, two different substrates, that's a bimolecular reaction, and it's defined by V equals K, concentration of A times concentration of B. Now, it's really important that we're not using kinetics to prove a reaction mechanism, but the kinetics can rule out alternatives which don't fit that kinetic data. So this is what a first order reaction looks like. It's constantly changing in terms of rate, but if we were to take the tangent or slash derivative at any point, you'll see that we can define it based on the derivative in respect to the concentration of A. This is also a really nice way that we can identify based on 100% as our starting concentration. Percent is 100%. At 50%, the time it takes to reach there is our T1/2, which is our half-life, or half-time, for the degradation of this reaction, progress of this reaction. So how do catalysts function? Well, they lower the activation energy for a reaction, which we looked at the very first slide, or one of the first slides. And the way they do this is they modulate the transition state. So all these reactions go through transition states, which have energetic barriers because they're high energy, unstable systems. And this is that peak in a reaction where we have our starting point, our peak here, and our products. And then the reaction rate is proportional to the concentration of a reactant molecule with its transition state energy. So if we reduce the barrier, we can get through that transition state much more quickly with the same level of input energy. In biological systems, we're not heating things up, we're exciting the system to get over that transition state. So we're just bringing the transition state down so we can move through it more efficiently. Now this doesn't change the equilibrium for the process because our ratio of energy for the inputs to the outputs hasn't changed. Reactants and products haven't changed. So the equilibrium doesn't get shifted, but the rate of reaction gets shifted significantly. And there's actually this nice equation, the Arrhenius equation, which relates activation energy to rate constant. And A, in this case, is the Arrhenius constant, and that's unique to each individual reaction. And everyone here is familiar with what K, delta G, R, and T are. So this is a really nice figure which shows the approach of the enzyme in B compared to the approach A of just raising temperature, what it looks like. So in A, let's say we start with, I'll go with green. Let's say we start here and at a low temperature. You'll see that we have a fixed degree of energy that is essential to move to the transition state, and at the lower temperature we have a fairly large energetic barrier. Now, if we increase the temperature, we don't change the energy required for the transition state, but we have increased the amount of energy available. So the delta between the starting point and the transition state has gotten smaller, and this increases the rate of reaction. Conversely, if we look at B, we aren't changing the energy of our products or the energy available in our system. And we're certainly not changing the energy of our reactants. We're not changing the energy level of our products. What we're doing is we're reducing that barrier through a variety of mechanisms, which we'll get into in the next lecture. But some of it has to do with just physical alignment to reduce the amount of random motion and excitement needed to get things to collide with the right orientation. So we're just closing that gap. So it's not a temperature increase, it's just reduction of the energy required to move into the transition state. Since we've been talking about transition states, there's a difference between delta G and delta G not. One has to do with the overall free energy change for a reaction and is related to the equilibrium constant. The other is related to the rate constant. So one has to do with equilibrium processes, the other has to do with the rate, and that is a really important distinction to make. So try to keep these two separate. So what equations define the kinetics of an enzyme-catalyzed reaction? Well, simple first-order reaction displays a plot of the reaction rate as a function of reactant concentration, that is a straight line. Well, enzyme catalyzed reactions are significantly more complicated than that because of having to interact with the enzyme on the way to the reaction taking place. And we're going to go down that rabbit hole quite a bit in a minute. What's important to distinguish for enzymes is that at low concentrations of the enzyme and substrate, the rate is proportional to the substrate, just like in a first-order reaction. But at high concentrations, the enzyme reaction approaches a zero-order kinetic because of saturation. Because the enzyme only has so many sites which the substrate can interact with. So you reach a point where the limiting factor is the concentration of the enzyme. And therefore, the concentration of the substrate no longer matters after a certain point. So this is just a linear slope of a unimolecular reaction in which compound A is converted to product P, and you see that as the reactant concentration A increases, velocity increases linearly. Now, this is what it looks like with the enzyme. As our substrate increases, kinetic behavior changes from first order to zero order. So initially, we'll have a similar to first order kinetic because we have a low amount of substrate concentration in which the substrate is limiting the speed at which the reaction can take place. But as we increase in our concentration, because the substrate has to interact with the active site of the enzyme to go through a rapid reaction, these ratios change, and we start to run into a physical wall where the substrate is now limited by the concentration of the enzyme available. And this, and the speed at which the enzyme can facilitate the reaction. And this gives us a theoretical Vmax for these reactions, which this approaches as we increase our substrate concentrations. It's not going to be linear like a unimolecular reaction. This is classic behavior of an enzyme-mediated reaction. So, let's explore this a little bit more. You guys have probably seen the Michaelis-Menten equation before, but we're going to spend some more time with it. So this is created by models produced by Louis Michaelis and Michaelis-Menten, or Michaelis and Menten is fine. And their model of kinetics with enzymes is predicated on the assumption that substrates interact with enzymes, forming enzyme-substrate complexes. This is the ES, enzyme-substrate complex. So the enzyme is E, the substrate is S, the enzyme substrate is ES, and product formed is P. And we have multiple equilibrium taking place in this. So what happens is an enzyme and substrate combine reversibly to form the enzyme-substrate complex. Once it forms, this is defined, this combination of the enzyme and substrate to form the ES is K1. This is our forward equilibrium, our first one. Once you've formed the enzyme-substrate complex, you have two possible fates. It can reversibly go back to just being the enzyme and substrate in the absence of any chemical reaction, which is usually minor, but this reverse is K minus 1. Alternatively, the reaction can take place in which the substrate is then converted to the product, and as it converts to product, it's released from the enzyme because it no longer has affinity for that enzyme because it's now a different compound. And that forward irreversible reaction is K2. So you have K1 and K2 as forward progresses through the conversion of a substrate to a product, and we have a single reversible step in K-1 where the enzyme substrate complex can dissociate without a chemical reaction taking place. Now, the enzyme substrate complex concentration remains constant through most of the enzyme reaction time course. You can see that here, where it's pretty darn flat. But in the early state of the reaction, which is shown, let me grab it here and blown up, you can see that there's quite a bit happening over time in concentration between the enzyme and the product and the enzyme substrate complex. So this is a busy slide, but everyone calm down, take a breath. It's not that bad. So this is the Michaelis-Menten equation. I used to have people memorize the derivation of it, and there's some value to that. There really is in terms of the understanding of where this comes from. But you guys can look that up if you're interested. I will not be testing you on the derivation of the Michaelis-Menten equation. However, you do need to be able to use the Michaelis-Menten equation. So, this is it. V, or velocity of the reaction, is equal to the maximum velocity of reaction, Vmax, times the concentration of the substrate, divided by the Michaelis-Menten constant, plus the concentration of the substrate. So, we have two new terms here. Everyone's got velocity down. Everyone's got concentration of substrate down. What is Vmax? What is Km? What do they mean as discrete variables? Well, let's start with Km, the Michaelis-Menten constant. One, it's constant for any particular reaction. And it's derived from rate constants. You can also think of the Michaelis-Menten constant as the concentration of a substrate that achieves half Vmax. So that's a neat shortcut, because if you have Km, you have the concentration of a substrate, which will give us Vmax, half Vmax. And so let's take a look at this here. Km is equivalent to the sum of K minus 1 plus K2 over K1. Well, let's look at what that means based on what we just looked at below. K minus 1 and K2 are the two possible fates from the enzyme substrate complex. It's the rate constant for what happens after the enzyme substrate complex forms. And then we have K1 in the bottom, which is our rate constant describing how much the enzyme and substrate like to associate with one another to form the enzyme substrate complex. What this means is that if you have a small Km, because K1 is large, that indicates a very tight binding. It means that the enzyme and substrate have a high affinity for one another and they want to form that enzyme substrate complex. Conversely, if you have a high Km, because you have a high degree of dissociation and a low K1, that means you have a low affinity for the substrate. Another way to think of this is that under actual Michaelis-Menten conditions, the Km is an estimate of the dissociation constant of ENS. That's really what we're looking at. So it just gives us insight into how well an enzyme associates with a substrate. That's what it is. Vmax is our other constant. It's the maximal rate of reaction. And we never actually achieve it in reality, but we can model to it. To actually achieve Vmax, you'd have to have all enzyme molecules, every single one, tightly bind to substrate, and that's not going to happen in reality, but we can get close. And as we increase our substrate concentration, to make it more likely that the enzyme will be interacting with the substrate, we see an asymptote where the line approaches our theoretical Vmax. So, realistically, this is not an overly complex scenario. We have two constants, which are simply defined by, in this case, K2 times the concentration of the enzyme, and Km, where it's just our ratio of things that dissociate from ES and the formation of ES. And then we've got concentration over substrate, and that's everything we got in the equation. We actually talked about this a little bit already, but it's worth touching again. When the substrate concentration is low, dragging things around again. When substrate concentration is low, we have that first order, pseudo first order kinetic. And then as the substrate concentration gets higher, it starts to become more zero order as we approach our Vmax. So it's called rectangular hyperbolic dependence of velocity on S, and it's characteristic. Michaelis-Menten is characteristic of enzyme-mediated reactions. A couple other terms that are used all the time. Turnover number defines the activity of a single enzyme molecule. So how fast can a single enzyme convert substrate into product? Well, the winner here is catalase. Catalase is the fastest known enzyme. It's very good at catalysis, hence its name, and it is our primary defense against peroxide. It's the reason that when you put peroxide on an open wound, it bubbles, because it's taking the peroxide and breaking it down into oxygen and water to prevent it from having toxic toxic effects on our cells. And that is capable, a single enzyme of catalase can convert 40 million molecules of peroxide per second. That's a lot of peroxide radicals getting converted to oxygen and water. But some are not so fast. So, as you look down the list here, DNA polymerase can do 15. Chymotrypsin can do 100. Lactate dehydrogenase can do 1,000. That's still a lot. It's not 40 million. It's a lot. Now this is interesting. The ratio. So remember, we've got Km, Michaelis-Menten constant, which is looking at how strongly an enzyme associates with a substrate. We have our turnover number, kcat. And the ratio of kcat to Km gives us insight into catalytic efficiency of an enzyme. So how perfect is an enzyme? And what this is interesting is because it gives us insight into how well an enzyme performs when substrate concentration is low, because you can always overwhelm an enzyme with a substrate and make a reaction happen. But if biologically we have low abundance of a substrate, we don't want to store it up necessarily because that takes a lot of energy. How do we make an enzyme function, or how can we evaluate an enzyme's function under low substrate concentrations? This is really a perfect way to look at it. So, let's see, even though catalase is incredibly fast, let me zoom in here. Even though it's incredibly fast, it has a relatively high Km, which means under low concentrations, it's not as perfect as acetylcholinesterase, which is awfully fast, but also has a much lower Km, which makes it a much more perfect, well, an order of magnitude more perfect. So it's all about your perspective and what you're looking at and what the conditions you are using to evaluate. So when you guys were in chem lab and you guys were doing Michaelis-Menten equations, you probably had to convert everything to Lineweaver-Burke plots to graph. Or at least I certainly hope you so. And the reason is because these are nonlinear, and if you want to apply linear statistics, you need a linear system. So all this is an algebraic rearrangement of the Michaelis-Menten equation so that it conforms to y equals mx plus b. That's all it is. So we've got our two axis variables, y and x. We have our slope, m, and we've got b, our intercept. And you can see that based on what we've got here. We have our slope, which is our Michaelis-Menten over Vmax. We have our y-intercept, our 1 over Vmax, and 1 over V and 1 over substrate concentration are y and x respectively. So not a hard thing to use. It gives us a lot of data because we can just simply look at the rate of reaction versus our concentration, and by simply taking the double reciprocal and plotting it, we can grab the slope, grab the intercept, and we can ascertain first the Vmax, then plug the Vmax into the slope, and we've got the Michaelis-Menten constant experimentally from taking velocity of reaction over a series of concentrations of substrate. Fantastic. Now, there's actually a slightly better way to do it. The Haynes-Wolff plot is a slight improvement over Lineweaver-Burke because it has a more consistent degree of reduced error across the system. It's still underutilized. Lineweaver-Burke overwhelmingly is used compared to Haynes-Wolff, but you should be aware that there are some other models which are slight improvements. Once again, it's a different rearrangement, slightly more complex, but it's still y equals mx plus b, and we can still, by plotting substrate concentration on x-axis and the substrate concentration divided by velocity, so it's normalized on the y-axis, we can still ascertain Vmax and Km without it being arduous. So isn't that cool? By just looking at rate of reaction over a couple different concentrations, we can ascertain how strongly an enzyme is associating with a substrate. And we can identify how quickly it can possibly function. Not bad. So, environmental influences on enzyme function. And I don't want to spend too much time here because there's a little bit of a duh component to it. If you change the environment with a protein that's finely tuned to interact with a very specific substrate, you should expect it to change its function. Correct? So, if you get an enzyme hot or cold, you will change its function, its rate, its ability to interact with the substrate. If you change its environment via pH, you have all those ionizable groups on the surface of the enzyme. Of course, you're going to be changing its function. So, I guess that's the point here. If we have all these ionizable side chains that determine the refinement of the tertiary structure and how it interacts with the substrate and interacts with its aqueous environment. And substrate often has ionizable groups that can be altered by the pH as well. So you should expect enzymes to function in very specific environmental conditions, whether that's pH, temperature, etc. And it will affect Km and Vmax if you go changing these things. So here's a quick look into it. just pH specifically. You can see that trypsin or cholinesterase really likes to function in the mid-7s, around here. That's by design. However, pepsin really prefers to be a little bit below 2. Pepsin doesn't function under the normal pH ranges. It prefers highly acidic conditions because it's digestive, right? And under acidic conditions of the stomach, enzymes should be able to function in there. So by design, it needs to be in an acidic condition to function. Whereas if you throw trypsin into that kind of acidic condition, it will have zero function. So all enzymes have very specific conditions in which they need to function under biological systems. This is just a simple indication of how temperature can drive enzymes. All kinds of extremophiles do require very high temperatures, very low temperatures, very high salt content, etc. But the temperature that we operate under has to be highly controlled so that we have that high percent efficiency of activity. In this zone, we don't want to be falling off the slippery slope of not having function from being too hot or too cold. So what can we learn by looking at how enzymes are inhibited? How it changes their kinetics? Well, enzymes can be inhibited both reversibly and irreversibly. Predominantly reversibly, but occasionally irreversibly. Reversible inhibition can often bind at the active site where the substrate binds, but it can also, an inhibitor can bind the enzyme at a variety of sites on the enzyme that are not essentially the active site, and exert their effects through altering the tertiary structure, which affects the access to the active site. So let's get into this a little bit. Reversible inhibitors can bind in a variety of ways. We have competitive inhibition, where an inhibitor resembles the substrate, meaning it competes with the substrate at the binding site for access to it. And often these inhibitors bind the site more aggressively than the substrate, but don't undergo reaction. So they just kind of sit there because they don't want to get released, because they don't become a product that then dissociates. You also have non-competitive inhibitors, which bind a different site other than the active site, and that conformationally changes the entire enzyme just enough where it alters the access of the substrate to the binding site. And then we've got and two variations on that. You've got the non-competitive inhibitor that's pure, and then you have a less than pure variation on it. And in reality, and what does that mean? What's pure non-competitive inhibition? It means that a non-competitive inhibitor doesn't alter the tertiary structure. And that's just an ideal. A vast majority of non-competitive inhibitors are going to be altering tertiary structure inherently, because you can't interact with a protein with something and not change its structure. It's going to happen either to a lesser extent or a greater extent, but it's going to happen. We also have the uncompetitive inhibitor, and it's really important to not confuse the uncompetitive inhibitor with the non-competitive inhibitor. An uncompetitive inhibitor will only bind the enzyme substrate complex. It won't bind the enzyme until the substrate is associated with it. So, if we were looking at Michaelis-Menten kinetics, and looking at a Lineweaver-Burke plot, and testing how the rate of reaction is changing as we change concentration in a double reciprocal plot. And we can do this experiment under three conditions. We can do it with no inhibitor, we can do it with a dose of inhibitor, and we can do it with a double dose of inhibitor. If it's a competitive inhibition, what we see is what we're looking at. And going back to look at this, if we have an enzyme and we have a substrate, you can interact like this, or you can interact with the inhibitor at the same site as the substrate would like to interact with the inhibitor. And this is our competitive inhibition at the binding site. Now, what's important to note is that in competitive inhibition, there's no change to Vmax. Vmax doesn't change. Just because a substrate doesn't have access to the binding site doesn't mean the maximum rate of reaction couldn't be achieved by adding much more substrate. So in a competitive inhibition, because it's competitive in the truest sense of the word, let's say we have a little bit of inhibitor. Well, probabilistically, if we just put 100 times more substrate in there, the inhibitor is going to be less effective because it's going to have less opportunity to interact with the binding site than the substrate. So you can overwhelm a competitive inhibitor with substrate. However, even though Vmax does not change, you can see that the addition of the inhibitor does alter the Km for these systems. This is a really nice example of a classic competitive inhibition, which is feedback inhibition in our metabolism, where succinate is our substrate. It interacts with the enzyme succinate dehydrogenase, SDH, giving us the product fumarate. Downstream, malonate, if it accumulates, can act as a competitive inhibitor by coming back here. Oh, I'm dragging things all over the place. By coming in here, oh, try that again, and blocking access of the substrate at SDH to shut down production of fumarate. And the more inhibitor that builds up, the less access succinate has to SDH. So this is a nice case of competitive inhibition that's also feedback inhibition metabolically. Let's look at pure non-competitive inhibition. So this is, remember, when the inhibitor binds a site on the enzyme other than the binding site, altering the way it can interact. And in pure non-competitive inhibition, we see no change to Km, but we do see changes to Vmax. Now, like I said, this is kind of an ideal, because in reality, you mostly get mixed non-competitive inhibition, or as I referred to it earlier, as impure, right? And basically you're seeing intercepts that are not on the x-axis, where you're having changes to both Km and to Vmax, because the reality is you're just going to be altering the enzyme's tertiary structure by interacting with it with the inhibitor. And then lastly, we have the uncompetitive inhibitor, which only binds the enzyme substrate complex. And here you can see that we are getting changes. Oh, geez, I'm going to figure out how to use this thing eventually here. The slope remains constant in the presence of inhibitor. So slope doesn't change, which means 1 over Vmax isn't changing. However, you do see a shift in our Km values because of these shifts with minus inhibitor plus inhibitor. But when you have these multiple parallel lines, you know you have an uncompetitive inhibition. And even though it's very, very less frequent naturally, you can also have irreversible modification of an enzyme via a mechanism called suicide inhibition. Penicillin is an excellent example of a suicide inhibitor in which it interacts with the enzyme, causes a covalent change to the chemistry in the binding pocket, permanently modifies that enzyme to where it can no longer function, and it has to be broken down by the body and rebuilt from scratch. So, penicillin's a phenomenal drug because of that action. But this is a relatively low frequency event compared to our reversible inhibitors. So, how can enzymes be so specific? Most of you have probably heard of the lock and key analogy, which was the original hypothesis to explain the specificity. The idea being you had this perfect key that aligned perfectly with this perfect lock, and the two just fit perfectly, right? Well, that just doesn't happen to be true. And we're going to get into the details of exactly how untrue that is in the next lecture. The new model, it's not that new, is the induced fit model. Now here's my jab I have to take. Most of you probably learned about lock and key in 100-level biology. It should be struck from the books. It's wrong. It's kinetically incorrect. The idea of lock and key is that the best fit is between the lock and the key. In reality, in the induced fit model, the lock is a decent enough fit to the key so that they can associate. And as they associate with one another, they both change. The enzyme changes conformation so that it becomes slightly different. And in doing so, it changes the substrate so that it is more capable of reacting, undergoing the chemical reaction that needs to take place. You have this overwhelming amount of chemicals, mass, in the enzyme, going to a lower state to drive this tiny thing that's associated with it to a higher state that mimics the transition. And as that's happening, it's binding that transition state with higher affinity than it does with the substrate itself. And that's the beauty of the induced fit, is the two come together perfectly to drive the substrate into conformation, which mimics the transition state, and that is how we are reducing the energy of activation through physical manipulation of the structure of the substrate. That's amazingly elegant. Lock and key is wrong, induced fit is possibly also slightly wrong, but it's way less wrong than lock and key. So, case in point, what I want people to take a picture of in here is that the glucose fits in neatly into hexokinase, but when they associate, you can see some conformational changes in which the glucose is actually buried, and the pocket, which is accessible before it associates, closes up and becomes inaccessible to solvent. So, significant conformational changes take place when they associate, which provides a more perfect fit between the two of them than existed before they were these separate entities. Couple last takeaways. Are all enzymes proteins? No. We also have ribozymes. Ribosomes have enormous amounts of catalytic potential as enzymes. They're segments of RNA which have a lot of catalytic potential. We also have abzymes, which are enzymatic bodies which have catalytic potential. So, simple yes, no. Are all enzymes proteins? No. There's plenty of enzymatic potential in ribozymes and abzymes as well, but a majority, in terms of diversity of enzymes, are proteins. And that's it for this lecture. So you can tell there's a lot to talk about in enzymes. This is a 1-hour lecture now. When we pick up the next one, it's going to be in terms of its function, the details of how enzymes function, now that we know how to describe their function, and the basics of what they are. We can get down the rabbit hole of how an enzyme functions, and then lastly, on a shorter lecture, how we regulate enzymes biologically. So, expect those to be up by the end of the week, and this should give you some material to dive into. So I'll talk to you all later. Bye-bye.